Genetic model organisms. The house mice is the model organism for humans.
Genetic model organisms are species that have certain qualities that make them fit to be used as models for genetic study. According to Jessica A. Bolkner, there are qualities that make a species a good model organism. They include rapid development, ready availability, small size or easy storage, easy to manage, and availability to analyze phenotypes.
Genetic model organisms were first used by Gregor Mendel, who is considered the father of modern genetics. He studied pea plants and how they pass on their traits to their offspring. Since Mendel's time, many other organisms have been used as models to study genetics and better understand biological processes of all life forms. Model organisms range from bacteria to mammals and help us study how human genetics works.
The mouse was first to demonstrate Mendelian's genetics in mammals by having the inheritance of coat color in mice observed by Lucien Cournot, a French biologist in 1902. The mouse is especially used as a model organism because of its qualities as a model organism and because of its genetic and immune homology with humans. Mice grow to about 10 centimeters from nose to base of the tail and weigh about 25 grams. They have a lifespan of 2 to 3 years under protective conditions. They have a gestation period of about 20 days, giving birth to 14 young, and they young grow to full adults in 6 to 10 weeks. With these characteristics, they are relatively easy to keep and manage. Their comparatively short life cycle makes them suitable to use in mammal genetic research with humans.
To exemplify the importance of the mouse as a model organism for humans, we analyzed the X-linked dominant disorder focal dermal hypoplasia, or FDH, and how it affects mice and humans. FDH is caused by mutation in a gene called PORCN. PORCN is a gene that codes for molecules that are involved in activating signaling pathways that promote embryo development and homeostasis of adult tissue. FDH is an inheritable condition that can only be passed by the mother since it is on the X chromosome. The condition is very severe and is lethal to all males in humans and mice due to the lack of another X chromosome to make up for the defective one. Although females do survive, the majority die as well from the severity of the syndrome. In humans, this disorder's phenotype is typically patchy and hypoplastic skin. It can also feature, among other mutant phenotypes, abnormal digits and kidneys, defective abdominal wall, and low bone density. FDH has comparative effects on mice, causing patchy fur and skin, as well as the other characteristic defects that humans have.
We also learned from mice that these effects arise during embryo development. By analyzing mice embryo, we were able to see how a normal and a mutant mouse differ in development. The one on the left is healthy, while the one on the right has the disease.
The reason the mouse is a good model organism for this specific mutation is they have the same gene on their X chromosome. As a result, research can lead to a better understanding of FDH and new treatment options for patients. The reason the mouse is a good model organism for this specific mutation is they have the same gene on their X chromosome. As a result, research can lead to a better understanding of FDH and new treatment options for patients.
Because of examples like FDH and many others, mice are an ideal model organism to study genetic disorders in humans. The kind of knowledge we can acquire from mice can help us better understand genetic disorders and learn how to treat them.